Hello everybody, my name is Charles Dwyer and I'm going to be presenting the leaching experiment that we performed in Transport Phenomena Lab. Leaching affects many industries including corrosion, waste disposal, and manufacturing. For our purposes we used a solid glucose sphere and we dissolved this in water using a reactor tank, and we tried to determine the mass transfer coefficient at two different mixing speeds, 60% and 100%. This is useful in the prediction of mass loss over a long period of time in the event that you don't have a long time to test something. Our group immersed the glucose sphere into the tank of water and mixed it with a small motor at two different mixing speeds, 60% and 100%. Water samples were taken at 4-minute increments so that the absorbance and concentration could be calculated and found from the spectrophotometer and Excel calculations. On the leftmost image you can see the reactor and the container for water that we used for this experiment. Next to it you can see the glucose sphere that we used. The packaging of the candy is right next to that. On the right side of the slide you can see that the motor speed dial, we turned this up to 100% for the first trial and 60% for the second trial, and on the right side you can see the spectrophotometer, which measured the absorbance and which we were able to calculate the concentration with in the Excel document. We filled the tank with 1400 milliliters of distilled water. Then we measured the dimensions of the glucose sphere and put the glucose sphere into the water. We then turned the mixer to full speed and used a slow-mo recording to calculate the revolutions per minute using a smartphone. We started the timer to calculate when every 4 minutes was up we would obtain a 5 milliliter sample using a syringe so that we could record the absorbance when we calculate it with the machine and the spectrophotometer. We then took that sample and placed it into a quartz cuvette to use in the spectrophotometer. After enough color is detected in the samples, we were able to calibrate the spectrophotometer wavelengths so that we could maximize the absorbance. We then recorded the absorbance value for all remaining samples and we graphed those into an Excel document. We then repeated that for a slower motor speed, which was the 60% trial 2, and then we cleaned up our lab space and performed the remaining calculations on the data in Excel to find the concentration and the mass transfer coefficient. Here are the two equations that we used the most in the Excel document. The first equation converted the absorbance into the concentration, and the second equation determined the mass transfer coefficient. On the left side of this results slide, you can see that the trial 1 table with time from 0 to 40 minutes and the absorbance is listed. For trial 2 on the right side, you can see that the time went from 0 to 64 minutes because it was a slower mixing speed and it took longer, and the absorbance is listed accordingly. The calculations that were performed on these values are shown in these next few slides. For trial 1, here are the absorbance, concentration, and then a few other calculations that we used to determine the mass transfer coefficient in those formulas. And here are the ones for trial 2, which obviously have more times, so there's a lot more information to go through in this section. Here are some graphs that show the mass dissolved over time for the fast speed, which was 100% for the motor, and the slow speed, which was 60%. You can see that the mass dissolved increased over time, which makes sense. As you can see, we were able to find the mass transfer coefficients with Excel. Now there are a few possible sources of error that we determined. One of them would be dirty glass in the spectrophotometer. In the event that you got fingerprints on the glass, you could have gotten a faulty reading. We also started with plastic cuvettes instead of quartz, which also could have contributed to any error. And the initialization was performed with the plastic hardware because at the time we didn't realize that that could have been causing error, and then we eventually later switched to quartz. So if we had initialized with the quartz cuvette, we probably would have gotten more accurate results. In conclusion, we did find that the experimental results can yield the mass transfer coefficients for both speeds. We also found that mixing speed does change the coefficients, which does make sense. Obviously, if you have something mixing at a faster rate, it should dissolve quicker. It's also important to use high quality cuvettes throughout the entire experiment, which would eliminate the error that we found.